All right, so today we're going to take our look at DNA replication. So DNA replication happens when cells need to make a copy of DNA, and we learned one of the most important times that is during the cell cycle, during the S phase. Remember the S phase, the synthesis phase, when we're going to synthesize new DNA before we go through mitosis or meiosis in making daughter cells or gametes. So DNA replication is the mechanism, the process by which we can make those additional copies of DNA so that cells can divide and create new somatic cells or new gametes. So DNA replicates based on what's called a semi-conservative model. Because base pairing allows each strand to serve as a template for a new strand, that each new double helix of DNA contains half uh, of its DNA, or one whole strand that is from the old double helix, so from the old uh, double helix of DNA that was supposed to be copied, and half of it is completely made from scratch. So let's look at this model. The blue might be, let's say, a chromosome that we zoomed up that we're copying during DNA replication in the S phase. Notice that the two new copies of the DNA both contain half that is right from the old DNA, and only half of it is made completely brand new. So each of the new DNA double helices contain half completely newly synthesized nucleotides and half old. That's why it's called semi-conservative, because it only makes half of the new strand from scratch. So what we need to remember before we go forward is that DNA again is antiparallel. So nucleotides in DNA are bonded from phosphate to sugar between the 3 and the 5 carbons. And here's a picture where the carbons are number 1, 2, 3, 4, 5. So look at how in this strand the 5 carbon in each one is closer to the top of your screen, and on the other side the 5 is close to the bottom of the screen, so one is just like upside down. That's what we call anti-parallel. Okay, so a DNA molecule has direction. 5 this way, 5 that way. We could flip it around, 5 that way, 5 that way. All right. As you can see, because of this, the complementary, so this strand on the right runs in the opposite direction as this strand on the left. That's important to remember because it will cause a complication as we go through this that will make DNA replication a little bit more involved than it would otherwise need to be if both strands were parallel, if they ran in the same direction. And there's just the end marker is a little bit bigger so you can see. So remember that hydrogen bonds link the two strands together, and these hydrogen bonds are relatively weak compared to the phosphodiester bonds that link the nucleotides on the side. The idea is that these hydrogen bonds are relatively easy to break compared to the much stronger phosphodiester bonds that join the backbone together. So in DNA replication, there's a large teams of enzymes that coordinate together to be able to replicate the DNA. So the idea is that what we're going to do is split the DNA molecule in half, so cut those hydrogen bonds, and to synthesize new complementary DNA to the old strand, kind of like in this picture. And then eventually we will get two new DNA molecules that can be used for the two daughter cells after mitosis or meiosis or whatever process is happening. So we're going to break this into 4 steps. The first step is we're going to unwind the DNA. So an enzyme named helicase, and this is going to be an important one to know, You can think of it as a pair of scissors, unwinds and parts or cuts the hydrogen bonds to kind of unzip the DNA into two separate single strands. So there's the helicase enzyme, that purple triangle. The single strands of DNA are stabilized by these yellow molecules are called single-stranded binding proteins. They prevent the DNA from what we say reannealing, coming back together right after it was separated. Okay, so here they are keeping the DNA from coming back together. And this site right here, right where helicase is actively unzipping and unwinding the DNA, we call that the replication fork. Okay, and then right on the other side of the replication fork, you can see what we call DNA gyrase, and that's an enzyme that prevents the DNA from tangling up and makes it a lot easier to unzip and unwind. Right, so in our first step, we have helicase that does the unzipping, our single-stranded binding proteins, which keep it unzipped until we can make new DNA in that space, and then gyrase that keeps everything untangled so that helicase can do its job. The second step, another enzyme which is called RNA primase, is going to add what we call a primer. It's just a small section of RNA to the 3' end. Here's the 3' end of the newly unzipped DNA. So here's the primase just adding that little primer. The reason that needs to be done is the main attraction to this show, this enzyme called DNA polymerase 3, can only add new nucleotides to an existing strand of DNA or RNA. So that enzyme requires this primer of RNA to get started. So primase is adding the primer. Look right here. Here's the unzipped, unwound DNA in the black, and the dotted red is that RNA primer that primase is putting down right there. So step 2, RNA primase adds an RNA primer to the 3' end of our DNA template. 3' end. Then the third step is really where the action gets going. We build our daughter DNA strand. So we go base by base or nucleotide by nucleotide and add in the matching or complementary DNA nucleotide going in the opposite direction. So DNA polymerase is the enzyme of the day here, and its job is to do just that, to move along the strand from the RNA primer going the opposite way, adding complementary DNA bases. So here it goes. This is our DNA polymerase doing its job. I note I didn't put the RNA primer in here. You can think of it's down beyond the bottom of your screen just to save some room, right? So it's going to continue up this way, and the DNA polymerase, unlike in this animation, would actually move along this way, actually move along, adding new nucleotides going up towards the top of the screen. So you can think about it as adding to the 3' end here. So the 3' end, if we go back, is this way. Right here is the 3' end, and so it adds new nucleotides onto the 3' end. The fourth step is we replace those RNA primers with DNA. So if you think if we're adding DNA, we're going to have those little chunks of RNA from the primase that's still stuck in there, and we don't want that. So a closely related enzyme called DNA polymerase 1, right? This one's called DNA polymerase 2. DNA polymerase 1 is going to come back and remove these primers, check them out, and put DNA in its place. So that's the basics of how DNA replication occurs, but we need to modify it just a little bit, because remember I said that because DNA is antiparallel, it's going to complicate things, and this is why. So here is a picture of the replication fork. 35. Our antiparallel nature, see the coming apart. Here's our single-stranded binding proteins in the black. And here's DNA polymerase working this way. From 5' to 3', starting from adding to the 3' end, going from 5' to 3'. DNA polymerase can only work in a 5' to 3' direction. So, on this bottom strand, which we call the leading strand, DNA polymerase, as long as it's got a place to start, can keep working towards the replication fork. And as helicase continues to unwind the DNA, it can keep going and going and going and not stop. On the other hand, though, DNA polymerase, if it was going to go this direction, would have to go from the 3' end to the 5' end of the new strand. That's not allowed. DNA polymerase, pretty straightforward. It can only work from 5' to 3'. So what it needs to do is to go backwards and synthesize the new DNA in segments. As more gets unzipped, it proceeds farther and goes back to where it left off. So we say that on this top end, called the lagging strand, DNA synthesis is discontinuous because it needs to start and make some DNA, and then, as more gets unzipped, stop, go further up and work backwards, and stop and work further up and go backwards. And each of these times, it needs a new RNA primer. So many RNA primers need to be placed on, this top strand called the lagging strand, whereas only one primer at the initial site needs to be placed on this leading strand on the bottom. So, like I said, DNA polymerase 3 can only build onto the 3 end of an existing DNA strand. It goes from 5 to 3. And so this lagging strand develops what we call Okazaki fragments. Here's Dr. Okazaki who discovered them. And those are those segments of where the polymerase had to pick up and start again, each interspersed with an RNA primer. Like we said, eventually DNA polymerase 1 goes back and replaces all of those RNA primers with DNA. But on this lagging strand, we need a special enzyme called ligase, which is like the welder to weld those backbones together, because otherwise they could just fall apart. So ligase only does its job on the lagging strand to fix the gaps between those Okazaki fragments from where DNA polymerase 1 removed the RNA primers. And there we have the little boxes with leading and lagging strand. So here we have another way to view it. So here was the leading strand on the bottom and the lagging strand on the top. On the leading strand, we've got primase that puts our RNA primer, and then DNA polymerase can continually synthesize new DNA, and eventually DNA polymerase 1 can come back and replace this RNA primer. On the lagging strand, though, RNA primase has to pick an initial site to begin the synthesis, and polymerase 3 needs to work backwards from 5 to 3 away from the growing replication fork. And primase needs to continue to put new primers as more of the DNA is unzipped and unwound. And so we have this discontinuous synthesis like you see here in the picture. So like I was talking about, after we have these Okazaki fragments, okay, each one of these, right here is an Okazaki fragment, okay, we eventually have to replace that DNA with RNA and then weld those together with ligase. So here's DNA polymerase 1, removes the RNA primer, removes the RNA primer, removes the RNA primer, and ligase joins those Okazaki fragments together. So we have a nice big continuous piece just like at the bottom. There's RNA polymerase 1 replacing the primer at the bottom. So here, lastly, we have just another picture just to make sure we understand what's happening. So here's the direction of replication, because here's the replication fork and helicase. We've got our single-stranded binding proteins here in blue, keeping keeping the strands apart so we can get in there and replicate. And we've got primase in the pink and DNA polymerase 3 in the slightly lighter pink. Notice right here we've got an Okazaki fragment on the lagging strand, and a continuous synthesis occurring on the leading strand from 5 to 3, 5 to 3. And then we've got ligase here welding together the Okazaki fragments as DNA polymerase 1 replaces the RNA primers. Okay, so that will be it for this video on DNA replication.